Hello class. So this is the second part of the series Citric Acid Cycles. And so we stopped at stage 2 of this catabolism of proteins, fats, and carbohydrates that acts as 3 stages of cellular respiration. And so what we are going to discuss now is the oxidation of the acetyl coenzyme A, which ends up producing these NADH and FADH2, which are electron carriers that will eventually reduce oxygen into H2O, and it will also facilitate the production of ATP. And so recall that I ended the last lecture talking about there are several steps within the citric acid cycle, and we are going to discuss each step. So just make sure we are paying attention to 1, the enzymes, and 2, the products that are being formed, and keeping track of where all of the carbons atoms within the molecules, because if we end up labeling one of the carbons for the acetyl coenzyme A, we should be able to track where that label is going throughout the process of the citric acid cycle. And so the first step is the formation of citrate, which uses the acetyl coenzyme A and reacting it with oxaloacetate. The special part of this reaction is it starts off reacting with oxaloacetate, and when you get done with all the steps of the citric acid cycle, you will end with a product of oxaloacetate. That's why I can start this process all over again. And the citric acid cycle is keep on being fed with the acetyl coenzyme A and producing the oxaloacetate that will end up reacting with the acetyl coenzyme A once again. And so this first step utilizes citrate synthase as the enzyme, and it has water that contributes, and what ends up being the product is the coenzyme A gets free, and then we have the acetyl group that ends up attaching to the oxaloacetate to end up making a citrate. So the first reaction of the cycle is the condensation of acetyl coenzyme A with oxaloacetate to form citrate, catalyzed by citrate synthase. The reaction catalyzed by citrate synthase is essential in aldol condensation involving a thioester, which is the acetyl coenzyme A, and a ketone, which is the oxaloacetate. And so the formation of citrate mechanism is by utilizing the amino acid residues. This aspartic acid is going to deprotonate this acetyl coenzyme A, and then you have a protonated histidine residue that was going to act more so as an acid in donating the hydrogen. Also notate that oxaloacetate is specifically oriented in the active site of citrate synthase by interaction of its 2 carboxylates with 2 positive charged arginine residue, which is not shown in this figure, but in order to trigger this actual reaction, the oxaloacetate binds first in a strictly ordered reaction sequence, and this binding triggers a conformational change that opens up the binding site for acetyl coenzyme A to happen. And so you're going to end up having this enol intermediate, and then from there you end up eventually creating this citral coenzyme A as it reacts with the oxaloacetate. And with that, you have hydrolyzation that happens and releases the coenzyme A, and thus having the free citrate for the completion of the first step of the citric acid cycle. And so the second step is the formation of isocitrate. And so although the equilibrium mixture at pH 7.4 and 25 degrees Celsius contains less than 10% of isocitrate in the cell, the reaction is pulled to the right because isocitrate is immediately consumed in the next step of the cycle. And so if you just pay attention to the Gibbs free energy, you see that it is positive. So this is not one of those spontaneous type of reactions. So that's why when it is created, it wants to eventually go back to that starting material. But in order to pull that reaction forward, that product that is being formed gets used up and pulled pretty rapidly and right away, because it will know within the reaction it will want to convert back to the starting material of the citrate. And so it uses the enzyme aconitase for this particular reaction. And so this enzyme contains an iron sulfur center. The iron sulfur center is in red, listed right here. The citrate molecule in blue, which is listed right here. And 3 cysteine residues of the enzyme bind 3 iron atoms. You can see that right here. 1, 2, and 3. The 4th iron is bound to one of the carboxyl groups of citrate and also interacts non-covalently with a hydroxyl group of citrate, which is a dash bond right here, and the carboxyl interaction with the carboxyl group of the citrate, and then a basic residue in the enzyme helps to position the citrate in the active site, which is shown right here. And the iron sulfur center acts in both substrate binding and catalyst. And so the third step is the oxidation of the isocitrate. So this is happening immediately. And so it utilizes the isocitrate dehydrogenase, which utilize a NAD plus, which gets reduced and thus can go on and make ATP as a electron carrier. And so with isocitrate, it forms this oxalosuccinate, and so you'll see this manganese ion in the active site interacts with the carbonyl group of the intermediate oxalosuccinate. And thus from there, there is a decarboxylation that is happening, and it still becomes stable with this manganese coordination with the intermediate. And thus from there, some rearrangement occurs, and then you finally have a alpha ketoglutarate that comes from the oxidation of isocitrate. Then from there, we have an oxidation of the alpha ketoglutarate, which utilizes an alpha ketoglutarate dehydrogenase complex, very similar to the pyruvate dehydrogenase complex. And so it utilizes these cofactors, coenzyme A with that free thiol group, NAD plus, and then thus producing NADH and the succinyl coenzyme A and also carbon dioxide. And so the oxidation decarboxylation, in which the alpha ketoglutarate is converted to succinyl coenzyme A and carbon dioxide by the action of the alpha ketoglutarate dehydrogenase complex. NAD plus serves as an electron acceptor, and coenzyme A is a carrier of the succinyl group. The energy of oxidation of alpha ketoglutarate is conserved in the formation of the thioester bond of succinyl coenzyme A. And this is favorable condition because we have a negative free Gibbs. And so the fifth stage is a conversion of succinyl coenzyme A to succinate. And so this utilizes an enzyme, succinyl coenzyme A synthetase, and utilizes the cofactor of GDP along with inorganic phosphate to end up generating GTP, and now we have the free coenzyme A with that free thiol group, thus producing this succinate. And so the mechanism behind this is, in step one, the phosphoryl group replaces the coenzyme A of the succinyl coenzyme A that is bound to the enzyme, forming a high energy acyl phosphate. And that is shown right here, that high energy compound. From there, in step two, the succinyl phosphate donates its phosphoryl group to a histidine residue of the enzyme, forming a high energy phosphohistidine enzyme, which is shown right here. And now you have that free succinate as the final product for this step of the citric acid cycle. And thus, when you have that GDP now, what that does is now it gets that histidine to lose that phosphate group, because remember, as a catalyst, where it starts out as, it needs to end. And so we need to have that histidine residue being back into its original form, and this formed GTP. But we need GDP again to keep feeding this reaction. And so in order to do that, GTP formed by the succinyl coenzyme A synthetase can donate its terminal phosphoryl group to ADP to end up forming ATP, which is shown right here. In a reversible reaction catalyzed by nucleoside diphosphate kinase, there is no change in free energy, shown right here, for the nucleoside diphosphate kinase reaction, and ATP and GTP are energetically equivalent. And so step 6 is the oxidation of succinate, where it utilizes the succinate dehydrogenase and uses a cofactor of Ph.D. that acts as an electron acceptor and thus producing a electron carrier that can go off to make ATP. And so the succinate from succinyl coenzyme A is oxidized to fumarate by the flavoprotein succinate dehydrogenase. And the enzyme contains 3 different iron sulfur clusters and 1 molecule of covalently bonded Ph.D. And so this particular step of the citric acid cycle can be inhibited by malonate. And malonate is an analog of succinate not normally present in cells and is a strong competitive inhibitor of succinate dehydrogenase, and its addition to the mitochondria in a laboratory blocks the activity of the citric acid cycle. And so the 7th stage of the citric acid cycle is the hydration of fumarate. And so this is a very stereospecific type of enzyme activity, as it produces a L-malate. And so what the special thing about this is that this fumarate, it has a trans double bond, as you can see from right here. And so it has to have this transform in order to have this particular enzyme activity to work. And this enzyme is called fumarase. And it produces this chiral center that is located right here that has the L configuration. And so this notate that when we do have the cis form, it will not create a D-malonate. So this is a very stereospecific type of enzyme activity that requires fumarate in order to continue on the reactions to end up forming this L-malonate. And so you have now on the next stage is the oxidation of malate. And so this oxidation uses L-malate dehydrogenase, where you have this cofactor NAD plus to act as a electron acceptor, thus producing this electron carrier that can go off once again and produce ATP, and then this finally produces the oxaloacetate that you've seen in the first step, and begin the process of the citric acid all over again and react with the acetyl coenzyme A. So in the last reaction of the citric acid cycle, L-malate dehydrogenase catalyzes the oxidation of L-malate to oxaloacetate, coupled to the reduction of NAD plus to NADH. The equilibrium of this reaction lies far to the left under standard thermodynamic conditions. As you can see, you have a positive gives free energy. But in intact cells, oxaloacetate is continually removed by the highly exergonic citrate synthase reaction. This keeps the concentration of oxaloacetate in the cell extremely low, pulling the malate dehydrogenase reaction towards the formation of oxaloacetate. And so this is just a general overview of all the enzymes that are involved in each of the 8 steps of the citric acid cycle, and you can see its thermodynamic properties of each of these enzymes and what is spontaneous and what is non-spontaneous and needs that pulling of the product in order to push the reaction forward. And so this citric acid cycle has intermediates that's always in flux. This oxaloacetate can also be used to make amino acids. Malate can also be used to make glucose. And breakdown of different amino acids can also make different intermediates of the citric acid cycles. And so at the end of the cycle, a molecule of oxaloacetate is regenerated. And citric acid cycle directly generates only 1 ATP per turn. And the 4 oxidation steps in the cycle provides a large flow of electrons into the respiratory chain via NADH and FADH2, and thus leads to the formation of almost 10 times more ATP during the oxidation phosphorylation. So when it does mention this citric acid cycle directly generates 1 ATP per turn, that step was what we seen way back here, where you have the actual product of succinate. That step, the 5th step, this 5th step is the one step that ends up generating that ATP, because we had that GTP that ends up transferring one of its phosphate group to the ADP to produce ATP. And then from there on, we have all this NADH and FADH2 as electron carriers that will do the 3rd stage, where it reduces the oxygen to be water and also facilitate the production of ATP. So overall, a single molecule of glucose can potentially yield around 38 molecules of ATP. And remember that ATP can be generated with glycolysis. Not only ATP is generated directly, but also indirectly by producing that 2-NADH, and then you have that 2-pyruvate to end up creating ATP as well via making 2-NADH, and then from there you have the oxidation of acetyl coenzyme A, which you have 2 of those, and that goes through the citric acid cycle and thus producing ATP this way as well. And this concludes the lecture on the citric acid cycle.